Well, everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we need to talk about the scientific method because I think there is a lot of confusion when it comes to how science actually operates. And hopefully we can clear that up today in this video. Thanks for being here. All right, so the first step in the scientific method is making an observation. An observation might be like you're seeing here. Hey, we've got some frogs in local waterways, and it seems to me that we have some chemical runoff in these waterways, and maybe the chemicals are having an impact on the frogs. And so that's an observation, right? That is thing number one. Now, that observation can also be through looking at other scientific literature. You read something and you observe, hey, you know what? They've found a problem. And so naturally, when you make that observation, you're going to ask a question. That question in our example might be, well, does the chemicals that are running off in these waterways actually have an impact on the frogs' lives and their survivability in that particular environment? And so we're asking a question here, but really important here. The question has to be answerable by science. It can't be moral or ethical questions because we can't set up an experiment necessarily to answer those. Now, it doesn't mean that we can't make moral or ethical observations based on the results of what we find in our science. But the question itself has to be answerable by science. This next thing is often highly confused, and that is, well, after we've made our observation, we've asked our question, now we need to form a hypothesis, and I am here to tell you that many people get this concept wrong. You've probably been taught that a hypothesis is an educated guess, and really in the sciences, that's not really correct. A hypothesis is a mathematical statement, and this is hugely important when we're actually doing science. And so I've got a couple of mathematical statements for you here. There are actually two different types of hypotheses. There's the null and the alternative. We'll start with the alternative. So we'll start down here a little bit lower. Alternative hypothesis is usually what we're thinking of when we design an experiment. And that is, hey, you know what? We've got this thing that we're looking at, and I think it's going to have an impact on our experiment. And so let me give you an example. Back to our frogs. We could set up two different experiments. We could set up an experiment where we have frogs in a pond that has no chemical. That is our control group. There's nothing changing about it. We're not altering anything. It's just frogs and just normal water. That is what we call a control group. There's no alterations. And then we can have our experimental group where we've got frogs that are in a pond, but there is some level of contamination. There's some level of the chemical. That's our experimental group. Well, the pond that has no chemicals would be data set A. And the pond that includes the chemicals would be data set B. And what we're saying is after we run the experiments, that the number of frogs that survive in each environment are going to be different. They're not going to be equal. That is the alternative hypothesis. We're saying that the frogs without the chemicals are going to survive at a different rate. They're not going to be equal to the frogs that are exposed to the chemicals. That's usually what we think of when we're thinking of a hypothesis. But there's another important side of that. The other mathematical statement, the opposite, is the null hypothesis, which states the two data sets are going to be the same. In other words, whether we add the chemicals or not, it isn't going to make a difference. Frog survivability in the control group without the chemicals is going to be exactly the same, statistically speaking, as the frogs that were placed in the pond with the chemicals. And so hypothesis is a mathematical statement. It's really not your educated guess. It's not what you think is going to happen. That's going to come next. It's either you run an experiment that supports the null hypothesis, or it doesn't support the null hypothesis. We either have to reject the null or we accept the null. So once you have that, then you can make a prediction. And usually when we're designing an experiment, when we're thinking about running an experiment, our prediction is going to be, well, we think the alternative is going to be true. Otherwise, why run the experiment? It seems like a waste of time. And so your prediction in this particular example might be, hey, you know what, we had this chemical, it is going to impact frog survivability. In other words, we think that the alternative hypothesis is going to be supported and that we're going to reject the null hypothesis that says the two data sets are going to be the same. This next part is perhaps one of the most time-consuming parts of the scientific method, and that is designing your experiment. Here, you want to control for as many variables as you possibly can. So in our frog example, we want to make sure The ponds are exactly the same size. The concentration of chemicals does not fluctuate when we're comparing them to our control. Are the frogs the same age? Do they have the same male to female ratio? Are they genetically similar? Are they the same species? Is there any other potential contaminant, any other factor that could contribute to our results other than the chemical concentration? We have to be very, very careful about that. So designing an experiment is huge. The other thing that we're doing here is we're trying to figure out, okay, well, how many replicates do we want to run? In this particular example, maybe it's not just going to be one control and one experimental group. Maybe what we want are multiple concentrations of a chemical in individual ponds. And so maybe we have a control, and then we've got 10 experimental groups, all with different concentrations of the chemical. And we want to see if there's a trend. The higher the concentration, the less survivability for the frogs. And so here we have to balance how many replicates do we want to run versus our resources, our time, and our money, because there's going to be a balance there. We can't just run infinite number of replicates because we'd run out of time and run out of money. And so that is the question here. The more replicates you can run, the more confidence you can have in your data. Well, after you've designed the experiment, of course, this is going to take a lot of time too. You're going to perform the experiment. One of the things I want to mention here is that often we think of this as an individual experience, and it's almost never that way. When you look at the scientific literature, you're going to see a lead author and then lots and lots of co-authors, because what usually happens is you have lots of people in a specific area of expertise that are going to contribute to your experiment. And so there might be a chemist there that is dealing with the chemicals, the chemical safety. You've got a biologist there who's dealing with the frogs. You have an ecologist there who's dealing with all the other things. You're going to have statistical analysis that we're going to talk about in a second. So you're going to have multiple people. It's going to be a team. Science is a team sport. You need multiple people. Not any one person is going to be good at all the different things that are required in a particular experiment. So performing the experiment, that lead investigator has got a lot of work to do because they're managing a group of people. And of course, you are going to collect a lot of data, and you're going to have to statistically analyze the data. It's not enough in the sciences just to say, hey, you know what? We ran these experiments. Yeah, you know what? It turns out there were some frogs that didn't survive at certain chemical concentrations, and therefore there's probably going to be some link. Not good enough in the sciences. You're going to have to statistically analyze that, and there are specific statistical tests that you will use depending on the number of replicates you have, the number of experimental groups that you have, and that has to be shown, your experiment has to be shown to be statistically significant. That gets back to our alternative and our null hypotheses. When you are statistically analyzing your results, you're going to either accept or reject the null. Sometimes people say you reject the null or fail to reject the null, but those are mathematical statements. Remember what I said earlier in the video, and that is what you're basing your results upon. And if everything goes as planned, one of the final steps is to publish your data, because if you run an experiment, you spend lots of time and money doing an experiment and you don't publish anything, well, you really haven't done it. So you want to go ahead and publish your data. And here, usually there's going to be a team of scientists that review your work and then deem it whether or not it's worthy to publish in their particular journal. And if it passes muster, they may sometimes have you go back and do some things differently, or take a look at how you've statistically analyzed your data to make sure that it conforms to their particular standards. And you publish your data, and then that allows other scientists to review that, use it for future experiments. Now, doesn't that sound just like the perfect scenario, this self-correcting science where people publish data, and then if there's ever a mistake, somebody else is just bound to catch it? Well, I'm here to tell you, it's not always the case. Science is not necessarily self-correcting. Usually what happens when people publish data in a peer-reviewed journal, there might be other scientists who are working on similar concepts. They might try to use part of the procedure or use some of the foundational information found in previous research to guide their own. And if it is not working, that may help self-correct some previously published studies. But a lot of times that is just not the case. In fact, what I tell my students quite often is, in this day and age, especially in this day and age, especially with AI, you need to be more discerning. Follow the money. In other words, what does that mean? It means scientists are people. People, just like scientists, just like all people, they have motivations. Now, when I say follow the money, that means who or what entity is funding the research? Can you depend on the research? And so, in other words, if you had a group of scientists, and this is unfortunately somewhat common in the nutrition space, if you have a group of researchers that somehow find that consuming high amounts of sugar does not lead to obesity, you should question that. And then you should look at, well, where was their funding coming from? Was it coming from a junk food manufacturer? Was it coming from an entity or an association that is getting their funds from some type of a junk food manufacturer? You have to follow the money. You have to see, where did the funding come from and what is the motivation for publishing the results that they published. Not saying that all science is corrupt. I'm not saying that at all. But what I am saying is there is a portion of it where you really have to question the results. You also have to remember that not all results are published. Okay, if you think that there's going to be, like in our example, the frogs and the chemical connection, well, if you find out there's no connection, that may never see the light of day. And so there's a lot of failed experiments, if you will, which I don't call them failed. If you have to support the null, I don't necessarily think that that's a failed experiment. You're still getting information from that, but it's not necessarily always something that's going to be published. You also have to understand the way science works. If you are a scientist, usually the way that you're getting money, because again, these experiments cost money, your livelihood costs money, so the money has to come from somewhere. It's either going to come from a governmental body or it's going to come from private resources. Either way, if you are performing experiments and never get anywhere in terms of publishing your results, you're not going to get money anymore. The government agencies aren't going to give you any money for what? Why would they give you money if you can't actually publish results? Private investment is not going to give you money if the results are not going to be publishable. And so there's a tremendous, tremendous amount of pressure that goes into publishing results, especially for professors who are trying to get tenure. If they're tenure track, it is really, there's a lot of pressure there. And so again, when it comes to the sciences, you need to follow the money. Do not just blindly follow the science, and do not just say, oh, the science is settled, or all scientists who hold this, believe this certain thing. Well, sometimes that is a self-correcting prophecy, and it's like, well, we'll just call all the real scientists the people who believe this, and anybody who doesn't believe this isn't the real scientist. So again, be discerning when you're going through the scientific literature. I know you're early in this process, but it is something you're going to have to be very, very aware of as you continue in the biological sciences. All right, everyone, so that is the scientific method and some of the potential pitfalls and downsides to the scientific method. As always, thank you so much for being here. Have a great week, everyone.